Yes.
Okay, so I think I will start. So the crux of my thesis is the systemic reaction. So how can a systemic reaction can trigger local regeneration? How regeneration can trigger a systemic reaction? So regeneration has fascinated the humankind for centuries. But what is regeneration? Regeneration is the ability to substitute cells, tissues, organs after an injury, an amputation, a disease, or because of aging. The concept of regeneration has been present historically, and so we can see it, for example, in the Aristotle's history of animals from the 4th centuries before Christ. The tails of lizard serpents, if they be cut off, will grow again. Actually, it was not well clear that serpents cannot regenerate their tails differently from lizards. Also, we can see this concept in the meat. And in particular, on the left, we can see the monster hydra. That is this gigantic serpent with 9 heads. And if you cut off one of these heads, the other one, the same will regrow. On the right, you can see the hydra, the real animal that actually has real regeneration abilities. This is an important model used in regenerative research. Here we can see the first draws of regeneration by Lazzaro Spallanzani by the 18th century. And in particular, we can see these rudimentary draws of salamander cell regeneration. So humans, as we know, have very poor regenerative abilities if compared with the other animals, for example, the fish, the zebrafish, and the amphibians like the axolotl. Here we can see the axolotl on the left and the zebrafish on the right that are real monsters of regeneration, as they have a very great regenerative abilities. So, and they are also the models that I used in my research. In particular, here we can see axolotl limb regeneration. So we can see how axolotl, differently from humans, can regenerate a complete limb after amputation. And zebrafish can regenerate the heart. Here we focus on heart regeneration. So it was demonstrated that heart regeneration is inhibited in the third mutant. And the third mutant is a fish missing the telomerase. The telomerase is important because it's an enzyme that adds DNA to the extremities, the telomerase of chromosomes. And this is fundamental because with each cell cycle, the telomerase gets shorter. And if the telomerase is not present, they will get shorter and shorter and shorter until they reach a critical length with DNA damage response, p53 activation, and cell cycle arrest, senescence, and apoptosis. And it also was demonstrated that p53 suppression promotes cardiomyocyte proliferation during heart regeneration in zebrafish. Here is more focused on the limb regeneration, and it is interesting that most of the studies have focused on local factors that can promote regeneration, limb regeneration in axolotl. This is an interesting study of a few years ago, in which it was demonstrated that after a limb amputation in axolotl, there is a systemic cycle activation, cell proliferation that regards not only the contralateral limb, but also heart, liver, and spinal cord. So there are many factors that can explain this systemic reaction. So we have, for example, hormones, or we have the innervation. And in particular, I was interested in the sympathetic nervous system. Also because the role of the sympathetic nervous system was very poorly studied. And the studies about it are very old. This is just an example of one of them that is from the 1978, and we can see that how new regenerates can respond to noradrenaline. So what is the main hypothesis? I was saying the crux is this systemic reaction, so how the system can influence the local injury and how the local injury can influence the system. And we have two main hypotheses for the two projects. So we have, can zebrafish heart regeneration trigger a systemic reaction, and vice versa, can the sympathetic system promote axolotl limb regeneration? So here I will focus a little bit more on the zebrafish heart regeneration hypothesis. So we can see that the hypothesis is complex. It is divided in three sub-hypotheses. So heart injury triggers cell proliferation, not only in the heart but also in distant organs, such as the gut, that was used as a model in my project. And then heart injury, the third mutant, for the reasons that we saw before, this should lead to a failure of cell proliferation. And then if you also suppress p53, this should be able to rescue the cell proliferation. So this was the main hypothesis of this project. Here we can see limb regeneration. So we can see the model and the hypothesis. So if we have, according to my hypothesis, if we have local amputation, then we have bleeding, and this bleeding causes a blood pressure decrease. And so we have a systemic response that triggered the sympathetic nervous system. And so the main mediator of the sympathetic nervous system are noradrenaline and adrenaline that interacts with the receptors. And in particular, I focused on the beta adrenergic pathway. So this pathway activates the adenyl cyclase that converts ATP in CAMP, and with the production of pKa that has been demonstrated to be involved in cell proliferation. So what are the materials and the methods? For the first study about heart regeneration, the plan was to use the wild type, the test mutant, the p53 mutant, and the double mutant test and p53 to perform the heart carrier injury, and then to study cell proliferation using the pCNA immunostaining and the apoptosis using the tunnel staining. And then to focus on the heart for the local regeneration, and on the gut to study, to look for the systemic effect. Here we see the approach of the other project about limb regeneration. So I use functional assays and an expression assay. For the functional assays, the plan was to have a double approach, pharmacological and genetical. And for the pharmacological approach, I use alpha and beta blockers to try to answer this question. Is the adrenergic receptor signaling necessary for limb regeneration? And for the genetic assay, I try to answer to this question. Is the CMP signaling important for limb regeneration? So I cloned the constitutively activated dominant negative gene AS that is crucial in the pathway, in the adrenergic pathway, in order to modulate the CMP levels. And also, I tried to clone a genetic biosensor to detect noradrenaline and adrenaline in vivo. So what are my data? Let's start with the heart injury. So here we can see that I successfully learned and performed the cryo injury. So this is a video of me performing the injury. I will quickly skip some seconds so we can see the time removing the skin. And we can see that here the heart beating. So I am exposing the heart. And then here the heart beating. And then I'm cleaning the side, and then I will perform a cryo injury using a probe that has been immersed in the liquid nitrogen. So here we can see this. So here we can see that my staining, the PCNA staining, unfortunately the staining didn't work. Also, I didn't have much time to optimize the protocol. It was only a 3 months project. Also tried the tunnel staining that worked only for the injured but not for the uninjured animals. So the next steps would then be to optimize the staining, the PCNA, the tunnel staining, and to answer the following questions. Is there a systemic cell proliferation after injury? Is this proliferation compromised if the telomerase is missing? Can cell proliferation be rescued if p53 is suppressed? So this is about the project about limb regeneration, the sympathetic nervous system. So we can see that there is, this is the pharmacological approach. So I use the beta blockers and the alpha-2 blocker. And we can see that there is actually no statistical difference in the blastema area between treated and untreated. However, there is morphology alteration that we can see as an effect of the alpha-2 and the beta-3 blocker. And here we can see that I have successfully cloned the dominant negative, the constitutively activated gene AS, and then I liposucted these constructs in axolotl cells. And we can see that the constructs are actually expressed. And here we can see that the biosensor that I cloned. So, okay, so it's a very complex tool. You can see that there is the adrenergic receptor has been used, and in the middle we have 2 different fluorescent molecules. This is because we can distinguish this way these 3 different states. If there is no fluorescence at all, we can see that we can say that there is no biosensor. If we see only orange fluorescence, we can see that there is only biosensor, but there is no ligand bind. But if we see both green and orange, with this tool we can say that we have the biosensor and the bind of the ligand. So what is the summary of this project? So it is likely that the propanol and the beta-3 blocker and the uridine don't block the formation of the plastema, and however, they can cause some blastema morphology alterations. I have cloned the dominant negative, the constitutively activated GNS, and in order to manipulate the CNP level in vivo, so a next step would then be to actually check if the tools are working or not. And I have designed I have cloned the human biosensor, but I was planning to clone the axolotl one, the chimeric, that is a combination of human axolotl genetics, in order to detect noradrenaline in vivo. So what is the outlook? What next? Is the cell actually cell proliferation compromised? Is compromised locally? Is compromised systemically? Can these beta receptor CNP pathway actually target the KGs in regeneration, such as sonic hedgehog or fibrogrowth factor? Is the alpha-1 adrenoceptor signal involved? Are the kinesis, the PKA, PKC involved? What about other monoamines? And it is possible in the future to translate these findings in mice and then in humans. I will just share again this slide that about the systemic reaction after the axolotl limb amputation, to say that the same lab at the end of last year actually demonstrated that the same lab from Harvard, the Witted lab, actually demonstrated that the adrenergic signaling is required for this distant cell activation after an amputation. So with this I will end. I would like to thank my supervisor, Professor Massimo Gulisano, for his support during this difficult period with the COVID. I always think that his advice is very precious and fundamental. And I would also like to thank Professor Tanaka and Professor Ferreira for the occasion to actually enrich my experience in their labs.